I have too much. Hello. Okay. Am I doing the right? Yeah. Okay. We didn't get that far. Yeah. Yeah, we were just starting 18. Okay, but I still need to do iClicker. So I haven't done iClicker.
Discussing chapter 19 before break?
Maybe. Probably not.
Probably not.
Probably not. Probably
I just-
19 will be
Because I
after
know
break.
that 19 is gonna be on the final.
Final. Correct.
So I didn't know if we were gonna cover that starting Thursday or.
Probably not, because we have to finish. We'll see. We'll see how it goes. Maybe.
I just was wondering because counting all the slides and stuff, we probably would, but I would really like to wait until after break.
Okay, stop doing it wrong. Okay. Oh, oh. Ha 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 ha. It's like I know that password was right. I did it for like the 6th time. Wasn't the password. Okay, iClicker's up. iClicker is up. All right. Okay, why didn't the Easter Bunny want to go out on Easter morning? Because he was having a bad hair day. I like that one. I like that one a lot. Okay, so you have checked in on iClicker, correct? I'm going to hide iClicker. Please check in on iClicker. Okay, I think today's like my favorite section about nucleotides. Some really, really cool stuff. So everybody got their case study. Sorry. So your case study and your questions, right? All right. How y'all doing? Y'all okay? No. I empathize if that makes y'all feel any better. And then I realized I have to write a grant over the break. So a grant. It's okay. Lots of things to do. All right. So we now know how nucleotides are made, right? We prefer to salvage them because energy wise, that's the smart thing to do, right? But we know we can synthesize them from scratch, right? We know how they're degraded. Now what we're going to talk about is kind of the dynamics of nucleotide metabolism, which is really the interesting part, right? So how do you get deoxyribonucleotides? So the 2' the difference between deoxyribonucleotides and ribonucleotides are the OH on the 2nd carbon. That's correct. So that's one way that DNA is different from RNA. What's the other way? Thymine instead of uracil in DNA, right? And that pairs with adenine. So the reason that we do that second one, right, is so that we can find spontaneous deamination events in DNA and get rid of them. So why do we not want to have OH on C2 of the ribose for deoxynucleotides? Like we understand why we have uracil in RNA and not DNA, but why do we not have the OH? So it's not as reactive. That is absolutely true. It's not as reactive. So if you have that hydroxyl group on the C2 of ribose, you have spontaneous cleavage of the phosphodiester backbone, right? And is that a good thing? So on ribonucleotides, RIBO, ribonucleotides, right? You have a OH on C2. Deoxy, right, is no OH on C2. So why? We said prevents spontaneous cleavage of phosphodiester backbone. This goes like all the way back to chapter 3 in the book, all the way to the beginning. So why is this good in DNA? And why would you actually want this spontaneous cleavage in RNA? The point of DNA is that it does not change, right? That it lasts. But RNA, tell me about RNA. Is RNA supposed to, do we want it to degrade quickly? Yes. Why?
So we can recycle.
The primary reason we want RNA to not last is because RNA is involved in gene expression, right? So you only want to make certain proteins in response to certain environmental cues. If those environmental cues go away, what do you want to happen? RNA to shut down, stop being made, right? And then any RNA that's still present will spontaneously degrade, and therefore that protein will stop being made, right? So it's all about responding to environmental cues. Whereas here, do you want the DNA to change in response to environmental cues? No, you want it to stay the same. Whole point is for DNA to stay the same. Okay, so good. No, it's not really in the notes, but it's really important to keep those big picture things in mind. Okay, so how do we make deoxyribonucleotides? You start from ribonucleotides. So it's a single reaction, one enzyme, ribonucleotide reductase. Ribonucleotide reductase is super insanely highly conserved across species. What does that tell you? It's old. It's ancient. It's so old, right? So if the sequence is conserved, the structure is conserved, which is true in this thing, and this ribonucleotide reductase, then we know it's really old, right? So this gives us the idea that how genetic material started was as RNA. Have y'all heard of the Rna World Theory? Yes, a little bit, yeah? Okay. So that's part of what we're going to talk about today. So we'll talk about why we think things started out as RNA, like everything started out as RNA and then changed. So this enzyme, ribonucleotide reductase, is the key that allowed us to change from an RNA world to a DNA world. And we'll see why in just a second. So what does it need? If it's a ribonucleotide reductase, if it's a reductase, it is going to require some kind of reducing agent, right? Like NADPH, like something that has hydrogens that are willing to give them up so that it can get those electrons that it needs. The electrons that it gets from NADPH are used to reduce a pair of sulfhydryls in the enzyme's active site. And we're going to look at the active site and we're going to look at the catalytic mechanism. And it's really one of the most unique catalytic mechanisms we have looked at across Biochem 1 and Biochem 2. It's really cool. So you start out with whatever nucleoside 5 diphosphate. So give me an example of what that could be. GDP could be ADP, ... right? Could be all of those. And what are you going to turn into? A deoxy. So you turn into D. GDP, DATP, ADP, excuse me, ... right? Okay. So let's talk about this RNA World Hypothesis, and then we'll go into details about the catalytic mechanism. So this theory was around for a couple of years before there was any evidence really to support it. And the first real evidence that said, yes, it could be that we started out with RNA as our genetic mechanism, is that RNA was found as a biochemical catalyst. So something that can function like an enzyme, but is really RNA. So if it's really RNA, but it functions as an enzyme, we call it a ribozyme. Give me an example of a ribozyme. Sounds really similar. It's an enzyme that catalyzes the transfer of mRNA into protein. A ribosome. So we know that RNA can function as holding genetic material, right? Messenger RNA. It holds genetic material, maybe not for long, but it does. We now know that it can function as a catalyst. So the more varied functions RNA can have, the more likely it is that it started out as the original blueprint for life, right? And then we were able to synthesize proteins from that RNA code. So we were able to look at the RNA, convert the RNA to proteins using amino acids, right? So all those functions could be done with just RNA. Is it the best? No. Is it the most efficient? No. But RNA could do them all. Technically, RNA can do them all. So here's what we think kind of started, right? So RNA was the first genetic molecule, right? And so then we have the ribozymes that were the biochemical catalyst, right? So this is the catalyst. So if this system is like it is, right? What's the problem? You just told me RNA is different than DNA, right? So what's the problem? It's unstable, and my RNA is going to change. It's going to mutate a whole lot more than it should, right? If we want it to be stable. So eventually what's going to happen is that this original enzyme, this original ribozyme, is going to make copies and mutate in order to turn into other things, right? So our ribozymes here, the genes that encode them, are going to mutate. They'll duplicate, right? Remember we talked about this in Biochem 1, that genes can duplicate. And then if you have a duplication event, the original gene can do its normal job, but then that new gene can accumulate more mutations and do a new job. Do you remember that? So that's what we think happened. And then if you duplicate it but gain mutations, now this is an enzyme that can convert our RNA into proteins, right? So that's one building block that was able to happen. But the other thing that also happened is that one of these ribozymes was allowed to mutate, got another copy, get some more mutation. And we converted that RNA now to DNA. So our ribozyme gene mutated, duplicated, mutated, and now is a ribonucleotide reductase. So it can take RNA and turn it into DNA. No, because this is just taking the individual nucleotides, whereas that is actually doing a whole strand. So it's different. It's very two very different enzymes. Okay, so now, do we want to keep up this part where we have an RNA template going to RNA copies? No, not really, right? Now we want to switch over and make our template composed of what? The DNA, right? We no longer want the template to be RNA because we have now gained an enzyme that allows us to make more stable nucleotides. When we put them together in a strand, they don't spontaneously break apart, right? So this is the transitional period where we can make deoxyribonucleotides, but we don't quite make DNA yet, right? So now, once we evolutionarily gained the advantage of being able to make DNA, it's more stable, it hangs out, it's a better genetic material. And so from DNA, we make the RNA copies, then from the RNA copies, we make the protein, right? This is today. Isn't that kind of cool? So it's all really based on evidence that RNA can do so many different things. And we know that RNA still has functions today, and there are other ribozymes that function in normal cellular mechanisms. But we don't talk about that. That's like at the very, very, very end of the book. And we don't actually get there. Okay. So how does this enzyme, ribonucleotide reductase, work? The whole point is to take some kind of nucleoside diphosphate and turn it into a deoxynucleotide diphosphate, right? So in order to do that, what you're actually going to have to do is add a hydrogen here on this one, and then release it as water. So when I said that it requires, what does it require? If I could fit it all on the screen, that'd be great. It requires NADPH, right? That's what we said in the beginning. It requires NADPH. But does the enzyme actually use NADPH? No. Just needs the electrons. And NADPH is the electron donor. And then we have to go through this series of reactions in order to transfer those electrons to the ribonucleotide reductase. Now, it's different. This pathway is different in animals than it is bacteria. But if you look at the structures of the intermediates that hold those electrons, structurally they look the same. So they're not the same gene, but they structurally look the same. So in animals, right, this is the main pathway that I want you to know, right, because we are animals. Our NADPH is going to donate those electrons to FAD. FAD turns into FADH2. FADH2 gives up those electrons to thioredoxin. Thioredoxin starts out as a disulfide, right? What happens to that disulfide when it picks up the electrons from FADH2? It's reduced, right? Now, these reduced sulfhydryls will react with the ribonucleotide reductase. It has what? A disulfide, right? So the reverse happens. Now the ribonucleotide reductase is reduced. And then our thioredoxin, we go back to having a disulfide. I'll see that? So we need this reduced ribonucleotide reductase in order to convert our base from being a nucleoside to being a deoxynucleoside. Okay, y'all ready to look at the mechanism? Possibly, maybe. Okay. All right. I know. It's really tiny on your paper. So like if you want to print it out big, that could probably help. I should have done that for y'all. I should have printed this out big for y'all. But the first thing we look at, remember we said in the enzyme, you need to have reduced sulfhydryls, right? So where are those reduced sulfhydryls? Right here. So this enzyme actually works as, you have to have both subunits, and then you have kind of a mirror image over here actually. So you have another R1 here, another R2 here. So you have kind of clamshell looking. So both sides function, but we're only looking at one side of the enzyme, okay? So here are the two sulfhydryls that need to be reduced, right? So those are two cysteines. Then you have another cysteine in R1, okay? It starts out as a sulfhydryl as well. Now, in the R2 subunit, you have something really, really unique. What is this?
A radical.
A radical. It's a radical. So cool, right? So you have this radical right here, and this radical is stabilized by an iron molecule, right? Because iron's plus, radical is minus, right? It's extra electron, it's a minus. Okay, so now what's going to happen in our very first step? So let me come over here so I'm not standing in front of everything. So this tyrosine free radical right here, right? It's highly reactive. So it is going to be reduced by the cysteine that's in R1 that's close to it. So that's the cysteine 439. Do you have to remember the numbers of the cysteines? No, no. Don't have to remember the actual numbers. You have to understand the mechanism. Like if I ask you questions about it, you have to be able to answer questions about it. Okay. So the free radical is reduced. So if the free radical is reduced, that means that that tyrosine picks up the hydrogen that was on the sulfur. But now what does that do to the sulfur? Now the sulfur carries the free radical, right? So what we're going to see is we're like passing the hot potato of the free radical, right? So that free radical is actually going to react, right, with not C2, but C3. When it reacts with C3, right, it's going to leave a radical on C3. So if we come down here, right, now where's the radical? On the C3, my cysteine, my sulfur on my cysteine is reduced, right? So I have my radical on C3. So what's going to happen with the C3 is the hydroxyl group on the hydroxyl group, they did this wrong. On C2, that's insanely huge. The hydroxyl group on C2 is going to gain a proton from one of the sulfhydryls that's sitting, that will eventually become a disulfide bond. Do you remember how I said in the beginning that the enzyme in the inactive form has a disulfide bond and we have to reduce that disulfide bond? This is eventually where we're going to make that disulfide bond. So I have to take away the hydrogens from here in order to make a disulfide bond, right? So this is the first place where I'm going to take away the hydrogen. This hydroxyl is going to pull a hydrogen off of the sulfur, right? So then we look here. So the C2 has pulled a hydrogen, right? This sulfur is negatively charged. This oxygen is what? Positively charged. And it kind of is stabilized by having this radical right here, right? So what's going to happen is that this isn't very stable, right? Because oxygen doesn't like having 3 bonds around it. So what's going to happen? The water is going to leave. The water is going to break. So you're going to break this bond right there. Water leaves. Now, carbon only has what? 3 bonds attached to it, right? So the carbon is positively charged on C3. I'm sorry, on C2. So C2 has a positive charge. C3 has what? It has the free radical. All right. With me so far? Okay, so now the hydrogen atom is going to be transferred from that second sulfur that's going to eventually participate in the disulfide bond to the C2 carbon. Now, what does that give the C2 carbon? A complete octet, but now it doesn't have hydroxyl on it. No, because it's just picking up a hydrogen. So in effect, now we're almost to the complete structure, right? And at the same time, this carbon that has the free radical is going to pull the hydrogen that's attached to the sulfhydryl down here to give this base the correct number of bonds around all the atoms, right? So if the one with the free radical picks up a hydrogen and the one that's the cation picks up a hydrogen, now we're all what? What is that? What did we just make? A Dtmp, right? Because we have on C2 a hydrogen instead of a hydroxyl. So we've completed our product. Now we just need to regenerate the enzyme. So what's going to happen is your DNTP is released, then thioredoxin is going to come in, and it is going to do what? Let me zoom out a little bit. Here. Okay, so if thioredoxin comes in, what happens to our sulfurs? We've got 3 sulfurs. What happens to them? If thioredoxin comes in with sulfhydryls, it donates the hydrogens to create a disulfide in thioredoxin. And what happens to the cysteines in my enzyme? They turn into sulfhydryls again, right? We had a radical, one was a radical, one was a disulfide. Yeah? And we've just regenerated our enzyme. Isn't that cool? I think that's one of the coolest enzyme mechanisms. Okay, good. All right, so if you're going to have this enzyme floating around that can convert any old NTP into a DNTP, how do you regulate how much of each individual nucleotide to make, right? Because you have, let's talk about our nucleotides. What are our purines? Pure as gold, right? So pure as gold is A and G. And then what are our pyrimidines? You want to know how I know pyrimidines?
Not A and G.
Well, not A and G, but what do you do to a pie? Pyrimidine, you cut it. You cut a pie. Yes, ma'am.
Back to the other slide, on step 6, the DN show
It's showing one coming in. This one is this. It's a new one coming in.
that's not the one.
It's not the one being released. This is the one being released. Does that make sense? Everybody on that? Everybody got that? What she asked was, right, if you made this DNTP, why are they showing this being released when this is a NTP? Because it has the hydroxyl. But what they should have like circled it, they should have shaded it something. But this base is a DNTP. this one is I'm sorry, this one is NTP, this one is a DNP. NDP.
You were
Ooh.
saying NTP.
Oh my goodness. Too many abbreviations. Too many abbreviations. Okay, good. All right. So what were we talking about? All right, so we're talking about our purines and our pyrimides, right? And who binds with who? What are our pairs? A, T, C, G. Do we have to worry about U? Where's U?
Or just RNA.
Right, just RNA, but binds to,
Oh,
right? Okay, so how do we regulate which one of these we need to make? Well, the ribonucleotide reductase has 3 sites, really. Okay, it has an enzymatic site. One is the enzymatic pocket. The second site is a regulatory site. So they call this the substrate specificity site. They call it the S site. And then they have a third site, and the third site is the activity site, or the A site. So this is activity, and then this is substrate specificity. So if I were to look at the enzyme, I think it's kind of important because you've got a lot going on in this picture. Where's the enzymatic pocket in this diagram? The catalytic site. Oh, that's right. They call this one the C site. So where's the C site? It's right in here, right? This is the pocket, the enzymatic pocket. That's the C site, or the catalytic. Kind of looks like a C, but they all kind of look like a C. All right, the substrate specificity site is a place where you can have allosteric binding. So this one right here is the specificity site. So what you're going to notice in each of these diagrams is that there is something different binding in the substrate specificity site. Y'all see that in each of the diagrams? What's in A? Dtmp. What's in B? Dgtp. What's in C? Either DATP or ATP. And then D is going to be different. Look at A, B, and C. Do you see a third pocket? What's the third pocket? Is the activity site. So here's the third pocket. What binds to the third pocket in A, B, and C? ATP. Okay, what's different in D? It's DATP in D. Okay, so I just kind of want you to look at the differences, and now we're going to talk about each one. Okay, so which question? Correct. It is a deoxy. That is correct. And we're going to talk about why that's important in just a second. Okay, so look at A. Let's start with A. In A, what we are saying is that we have DTTP in what site? In the S site. The S site is where we're going to allosterically regulate the activity. So if we have DTTP, what we're going to do is we are going to promote manufacture of DGDP. Does that make sense? If we have DTTP in the S site, that means we're going to promote manufacture of DGDP. So if we have T, we're going to make G. If we have T, we're going to make G. If we have lots of pyrimidine, we're going to make some purine. So we're going to balance purine and pyrimidine. That makes sense, right?
It makes sense, but why would they not, like, I mean, I know we can't change biology to make it easier for us, but why not just do it like? Yeah.
Well, wait, just wait. Just wait. What's that?
Well, it wouldn't make a whole lot of sense because if you, like, if T's made more C's, you're trying to force the opposite to. Right, like, aren't you trying to force the opposite to what you want that to regulate?
That's exactly, just as many pyrimidines as you have in the DNA, you're going to have that same percentage of purines.
Gotcha.
That's how DNA is constructed, right?
That makes sense.
Okay, so let's look at the next. Well, before we do that, if you have DTTP in the site, you are going to promote DGTP, but you're also going to have an inhibitory effect on what? DCDP and DUDP. Because those are both also pyrimidines. So you're going to promote purines while suppressing pyrimidines. Okay, so now let's look at B. I'm trying to keep everything on the screen. Okay, if we have DGDG, oh my goodness, DGTP in the S site, what's going to happen? We're going to promote DADP. And at the same time, we're going to inhibit DCDP and we're going to inhibit DUDP. So if we have if we have G, what are we going to do? Promote A and inhibit what? C and U. Because what you want to have is all 4 of your nucleotides in the correct amounts. Okay, so you're not always going to be making a purine and then inhibiting pyrimidines. It's more complicated than that because you also have to have the correct ratio of your matches. So you just have to know them. You just have to know them. If you know the ones that promote, that's the really important. It's a really important part. There's only 3. There's really only 3. Okay, so what happens if we have in the S site DATP or just plain ATP? Doesn't matter. In the S site. So we're going to promote 2, DCDP, and we're going to promote DUDP. So if we have A, I can't fit them quite all. If we have A, look up at the top, we're going to promote C and U. Do you see how those are the only 2 that weren't promoted in the past? We're going to promote C and promote U. So what is the only nucleotide that we have not increased expression of or inhibited for that matter? T. I'm going to show you there's a different mechanism for making T. I'm going to show you what that is, but not yet. So in effect, everything but T, we have either inhibited or promoted, right? So that we get that complete balance of all the nucleotides that we need, minus T, because we haven't finished talking about T. Now, we got to talk about that activity site. What do you notice in A, B, and C about the activity site? If you have ATP in the activity site, what happens? So here, here, and here in all three. If you have ATP in the activity site, you can, the enzyme is what? Enzyme is active. Okay, now what happens if we put DATP in the activity site? And that is only here. Everything's inhibited, which means you are doing what to the enzyme activity? That's why we call it an active site versus a specificity site. The specificity site regulates based on which nucleotide binds, tells you which nucleotides you're going to make. Whereas the activity site, it's on or off.
Instead of when it's
It's a switch. What is what? Does that help?
Yes, a lot.
Mhm. Okay, so the last one we didn't talk about, right, was T. So what happens with T? So thymine is a purine or a pyrimidine. It's a pyrimidine. Okay, so it's a pyrimidine that's formed from methylation of uracil. So if you're regulating up here, if you are regulating UDP, what are you in effect regulating? T. If you're regulating U, you're actually regulating T. It kind of makes sense, right? If we started with an RNA world, did we start with T? No, but then when you transition to DNA, there became a problem, right? And so conversion of using U, T and DNA instead of U is a workaround for that. It would make a whole lot of sense if we started out as an RNA world. Okay, so here's our two structures. DUMP, it's hilarious, right? Don't call it dump. But it is DUMP. What's the only difference between DUMP and DTMP? The methyl group, that's the only difference, right? Because you have to worry about uracil deaminating, right? That's the worry with uracil. But the only difference that stabilizes that is that TMP has the methyl group. So how do you convert DUMP to DTMP? You're going to use the enzyme thymilate synthase. So thymilate synthase, right, is going to require a couple of things in order to work, right? If we're going to add a methyl group, we're going to need a carbon donor, right? So we get that carbon donation from serine. So you start out with dihydrofolate. You know how folate's really important in nutrition for nursing mothers and all that kind of stuff, right? The milk's fortified, and, you know, all that kind of stuff. That's because it's required in DNA synthesis. So you have dihydrofolate. Dihydrofolate, right, goes through dihydrofolate reductase. And this is going to be really important to know this mechanism because we're going to talk about it for anti-cancer drugs. We're going to use our electron donors of NADPH, right? We're going to pick up that extra hydrogen. So that's one of the hydrogens that we're going to need in our methyl group. Then we're going to use the serine and convert that tetrahydrofolate into a methyl tetrahydrofluorate, folate. And so this is where our carbons and our hydrogens are going to come from. when we turn DUMP into DTMP. For anything, any organism is at the stage where they need to replicate DNA a lot, you're going to need folate, right? So in pregnancy, in nursing mothers, right, the babies are growing and growing and growing. So you're going to be replicating DNA a ton. Okay. So if you need DUMP to make DTMP, how do you get DUMP? Right? That's the question. All right. So there are a couple of different ways. There are actually 3 different sources for DUMP. Here's DUMP. This is our goal that we looked at on the last slide. The last slide reaction we looked at was right here. DUMP to DTMP. Right? So this reaction is exactly what we saw on the very previous slide. So there's like 3 different ways that we can get there. Okay. So if we have DUTP, right? So we have the triphosphate. If we have the triphosphate, we can simply remove one phosphate with a phosphohydrolase, and now we have DUMP, right? That one's okay. You could also deaminate cytosine, right? So we have DCTP, we deaminate that to DUTP. Now we can convert DUTP to DUMP. So here's our first one. Here's our second one. The third thing that can happen is that we can phosphorylate deoxyuridine. So this is 3, deoxyuridine. So if I phosphorylate deoxyuridine, what is deoxyuridine? It's got the sugar and the base. Does it have any phosphate groups? What? It's a nucleoside, right? So all you have is the sugar and the base, and all you have to do is phosphorylate it, right? So this is sugar plus base only.
So this is our third source.
That's our third source. That's our third source. So it's really important that we don't accumulate DUMP. If we accumulate too much DUMP, if this gets too high, what do you think might happen? DUTP. If we get too much DUTP, what's the use for DUTP? What do we use it for normally? Make RNA. Okay. Now I'm telling you that we have so much DUTP that RNA doesn't need it anymore. Where might it go? Incorrectly. It's not supposed to go there. But if there's so much of it and you don't have enough DTTP, where will it go? If this gets too high, you now have more DUTP than than DTTP. What happens? Well, eventually, yes, right? That could be the answer to so many different things. That's a great answer, though.
Yeah, really. I'll take liver for 500.
Right, it's either the answers in this class are either liver or cancer, right? Wait, I thought I heard somebody say, well, like, okay, you've RNA doesn't need it anymore. RNA is out of the equation, right? RNA is using all of the DUTP that it needs. What is it structurally similar to? DTTP. So it would actually get incorporated into DNA. It would incorrectly, incorrectly, TLY, incorporated into DNA. And that's bad, right? Why?
Because DNA is not supposed to have uracil in it.
Because then you have repair mechanisms that are going to go into overdrive. It's going to be really, really terrible, right? So this conversion right here, this step number 1 is super, super important. This enzyme prevents this from happening. So you don't have that misincorporation because you can convert DUTP to DUMP. Now that it's in the monophosphate form, can it be incorporated into DNA, into anything? No. Right? Now you're basically telling it that it has to go and make DTTP. But you're destining it to go and be made for DNA.
Which can then be properly incorporated.
Correct. Does that make sense? It's kind of complicated. It's kind of, you kind of have to sit down with this and explain it out loud with a dry erase board and some friends with an open book to help correct you as you go, right? Okay. All right. So now this is on the cancer kick, right? As we're talking about cancer. So inhibitors of our thymilate synthesis, these are all examples of anti-cancer drugs. So let's look at our kind of pathway in general before we add any drugs to it, right? So don't look at the minuses, don't look at stuff like this, right? So normally we have DUMP that's converted to Dtmp via thymilate synthase.
Which means there's no ATP. Like no ATP doesn't. Because the synthase not synthase.
Yes. In this case, yes. Not always. Not always. All right.
Exceptions.
Yeah, exceptions. So this is the same pathway that was just a couple of slides ago, right? Where in order for thymilate synthase to work, you need the dihydrofolate. The dihydrofolate uses NADPH to convert to tetrahydrofolate to the methyl tetrahydrofluorate, right? And this one has a methyl group that gets donated to UMP to make TMP, right? So this is the same pathway that we saw like 2 or 3 slides ago, however many that was, right? Okay, so what have we talked about before? We talked about 5-fluorouracil before, right? How does 5-fluorouracil work as an anti-cancer drug? It's a uracil-based compound, right? So it gets metabolized into this substrate that looks like UMP, but isn't UMP. It's an analog. So metabolized to analog that binds thymilate synthase. Yeah. Yeah. Okay. All right. What you could also have is an inhibition at any other point in this cycle. If we have things like methotrexate, that's a really, really popular anti-cancer drug. So methotrexate does not directly work on thymilate synthase. But what does it work on? Dihydrofolate reductase, which prevents the acquisition of those electrons from NADPH. If you can't pull the electrons from NADPH, are you going to make tetrahydrofolate? No. So it acts as an inhibitor. Aminopetrin is a similar drug. It does a similar job, right?
So these prevent the pull of the electron from.
It inhibits the enzyme that facilitates the transfer of the electrons in hydrogen.
Or is it the 5-fluoro?
The 5-fluorouracil is metabolized to this analog. So this FDUMP is the analog. So you have to think about which cells are going to need to replicate their DNA a ton. Cancer cells. Cancer cells, that's all they care about. They don't care about doing their function. They don't care about anything else other than replicating. So these anti-cancer drugs will kill cancer cells, but what do they also kill? Any healthy cell that is undergoing rapid cell division.
Like the stomach lining, hair follicles.
Like the stomach lining and the hair follicles. So you have hair loss and nausea with patients who are undergoing chemotherapy, right? Okay. So let's talk about the structure of some of these, right? I just kind of think this, if you ever want to get into like medicinal chemistry on designing anti-cancer drugs, this is really, really cool. Okay, so if we look at DUMP, right? If we look at 5-fluorouracil, what do we see? A very common structural homology, right? And so this 5-fluorouracil metabolized to 5-fluorodeoxyuridine. So this is what's presented to our enzyme, and can our enzyme use this? No, right? It's got this fluorine on here. That's not going to fit in our enzymatic pocket. It's not going to work, right? So it's going to inhibit our conversion from DUMP into Dtmp. Okay, so look at B. Dihydrofolate. Here's another anti-cancer drug. So this anti-cancer drug is very, very similar in structure to dihydrofolate, right? We're missing a nitrogen here. We're missing a nitrogen here. We're missing a nitrogen here.
The drugs are on the right side.
The drugs are on the right side, yes. I have to stand. My left and your left are different. We produce dihydrofolate. These are the natural, let me circle them. These are the natural substrates, natural substrate, natural substrate. And then we're looking at structural analogs that can be used. So look at B, both B and C are structural analogs to dihydrofolate. Right? Really, really similar, really similar. There's one drug, this drug down here at the bottom, trimethoprim. Trimethoprim is really kind of interesting because it's a different kind of drug. This is actually not an anti-cancer drug. So this is kind of put a little line. This is a different kind of drug. This drug is actually specific. Do you remember how I told you there were differences in animal and bacterial, the bacterial enzyme, right? You had different substrates that were required. It's highly conserved, yes, but there are differences. And so this one actually binds like 30,000 times tighter, tighter to the bacterial enzyme than animal. So what could we do? And it's the enzyme is the hydrofolate reductase. So what can we use this one for? Do we want to use this for anti-cancer? No, what do we want to use it for? Bacterial infections. It's used for UTIs. It's a very common drug for UTIs. Isn't that kind of cool? You can take advantage of differences. Like if you are a structural biochemist, you look at drugs that are very similar in structure, but induce these insanely different effects, maybe from one organism to another and all that. I just think this is super interesting. If I could do like another PhD, that would be really cool. I'd love to do one in that. I'd love to do another PhD in like marine biology. I feel like that would be really cool down here too. Sadly, there's not enough time in the day. Okay, so before we do this, you put somebody on chemotherapy, right? Let's just say you put somebody on chemotherapy. A lot of times it will slow the cancer, but sometimes it doesn't always completely kill the cancer. Why? Sometimes the cancer can come rearing back. Drug resistance. Let's talk about that. So now you have to think very specifically, biochemically, about why would cancer cells become resistant to these super toxic drugs that you were putting into the person's body. There are actually 3 different ways that this can happen. We talked about one of them when we talked about penicillin. What does the bacteria do to become resistant to penicillin? So the gene that codes for the enzyme that the drug binds to does what? Mutates. So you have mutation in target, we'll say enzyme target. And so now you have reduced binding affinity. For the drug. Right? You got to kind of think about bacteria and cancer as having some commonalities. Isn't that kind of weird? But it does. Okay, what's something else that you could do if your sequence stays the same? We're going to assume then that number 1 didn't happen, and we're going to, what's another way? Sometimes cancer cells become resistant to not just one drug, but like a plethora of drugs. Why could that happen? We talked about this in Biochem 1 when we talked about transport. Briefly, very, very briefly. So not just a channel protein, but an actual pump. So there are pumps that are designed to pump out toxins from cells, right? This is a good thing. You don't want toxic things building up inside of your cell, right? But cancer cells look at drugs, anti-cancer drugs, as things they need to get rid of. So they upregulate, you could say, membrane transport pumps. That is multi-drug resistance. Do you see how the first one multi-drug resistance? No, it's only one drug, right? It's a very, very specific drug. Okay, there's a third one that's also specific, kind of like the first one. If you keep making this necessary enzyme, right? And you give somebody a dose of drug, and that dose of drug is effective, right? Reverse. Upregulation. So if you get treated with so much drug will treat cancer based on the number of proteins that are present in the cell, right? So if I could increase the number of proteins that are floating around in my cancer cell,
You don't have enough drug to get rid of all the proteins.
correct, I'll run out of drug and then I'll have enough protein. So the most effective way to do that is not just to increase transcription. Increase of transcription is not going to be enough. You actually have gene duplication. So you increase the copy number of gene target / enzyme. Sometimes they call it gene amplification. Depends on what you're reading. But these are 3 different ways that cancer cells can become resistant. So if there are multiple ways that a drug can be resistant, do I want to, I have this patient coming in with cancer, do I want to just give him one drug? No, right off the bat, what do I want to do? I want to hit them, and I want to hit them hard, right? It is going to be hard on their body. Yes, it's going to be terrible. But at the same time, if you kill the cancer cells, that's the name of the game, right? So what we do typically is give someone a combination therapy right off the bat. So, see if I can do that. So sometimes we're going to give them a specific drug. This is the specific drug that we were talking about, the Thymilate Synthase inhibitor, right? One drug that targets a particular pathway that's important for cancer cells, like DNA replication, right? We're also going to give them a multi-drug resistance protein inhibitor so that it can't pump out this drug that I just gave it, right? Then I'm also going to give it an oncogenic kinase inhibitor. Remember how we talked about kinases being the enzymes that turn things off and on, right? So what's an oncogene? An uncontrolled, something that once it's going, the cell cycle goes go, 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 go. So this controls the cell cycle. So I'm basically going to shut down the cell cycle, right? Okay, and at the same time, I also want to give my immune system a better target, right? The immune system is supposed to take care of cancer cells. It's supposed to kill cancer cells. If it does not, then that's really bad for the body. So what we can do is we can actually take out cancer cells from patients and generate antibodies specific to those cancer cells, or you can take an antibody that's kind of a general cancer antibody, because certain cells certain cancer cells will display certain extracellular receptors, right? So they could activate the cell death pathway if they're active, like the caspase cascade. Remember the caspase cascade that we touched on real briefly? But it can also promote immune cell recruitment. So we want to hit them with everything all at once and knock out the cancer right away. So how's your patient going to feel on chemotherapy? I mean, there's no way around that, right? They're going to feel terrible, but it's absolutely worth it. And this combination therapy is not just seen in cancer treatment. We also use it in HIV treatment.
Always tell patients to stay inside more because their immune systems are compromised.
Well, so why is your immune system compromised? That's something different. Your immune cells are rapidly dividing cells. So just like visibly we see hair loss, visibly we see nausea. But
because we're doing chemotherapy, which targets that rapidly dividing cells, it's going to also target your immune system.
correct. It's just not a visible phenotype. Okay, so HIV treatment, same sort of deal. You're going to give the HAART, the heart therapy. So this is 3 different classes of drugs for HIV all at once. So you give inhibitors to all of the processes that viruses need, specifically HIV, right? HIV is what kind of a virus? A retrovirus, right? So if you can inhibit the integrase, right? Because what does HIV do? It takes its genetic material and does what with it? Turns it into RNA, then DNA, and then what does it do with that DNA? It integrates it into your genome. So if you can inhibit the integrase, then you can prevent HIV from working. But at the same time, in order for the virus to be reassembled in the cell, right? Once you make all these viral components, you have to have proteases that will kind of chop up things and make them active. And so if you can inhibit the proteases, that's going to stop the virus from replicating. And then other drugs that target different places in viral replication. But we don't want to just give one. We're going to give them all, right? So that gives the survival rate 30 to 50% longer than somebody who is untreated, right? So, all right, that is the end. That is the end. Okay, so we have 10 minutes left. Not as bad as cancer therapies, no, because all of these are targeting viral things, right? So do we really need integrase for our cells to work? No. Oh, yeah. Yeah, not saying that there aren't any side effects. There are, of course, side effects, but not anywhere near. Like you can look at a cancer patient and know they're a cancer patient. You cannot look at a person who is undergoing HIV treatment and know. You can't.
Unless they tell you, right?
Well,
But they would.
you can't
really
look
disclose.
at them.
No, that's
You
what I'm
can-
saying, like, unless they wouldn't tell you that, so you really wouldn't know. Right. I wasn't agreeing with you.
All right.
I didn't realize it sounded so not agreeing.
Okay, so we have 8 minutes. Next time, what's the plan for next time?
Case study.
We have one more case study. And then what we'll do is the next class period, we'll do case study, and then we'll use the rest of the class time to do questions, the discussion questions and stuff. And then when we come back, what happens when we come back?
Test.
First day that we come back.
Oh, first day of the Monday?
Well, no, I meant the Tuesday.
Oh,
I meant the
test.
first day that we're here, the test. And then we have chapter 19,
And the day before the test or homework's are due.
and then we're done. Yeah, day before test. So Monday, homework is due for chapter 18. So all the grades should be updated, except I apparently did not enter case study 2. So I will go down to my office. I did?
Yeah, we just never got it back.
You
Oh,
just
no,
didn't
I
get
don't have it.
You don't have, okay.
I told you.
I will go. I will bring them next time.
I literally just don't have it. So
I
mine.
tried.
You just don't like me, Dr. B.
I wanted to ask you if you would actually do class on Zoom Thursday. I could just email you that please send questions.
Yeah, email before and send me your questions.
Okay.
You know?
That's why I was telling her.
Yeah, I haven't done stuff like that.
Yeah.
Thank you.
Me, so
I have to submit like my slides today. So
Yeah.
nothing related. So I wrote the dates wrong. I thought the abstract was due today. And so I was like, oh.
Well, it's gonna be a draft.
It's gonna be a rough.
And then you and I need to look at my calendar and book 2 appointments back to back so that we can like go through your seminar.
We have to practice it. Yeah.
Because you want us to go over it together. Like, if I can give you feedback and you can make me things that need to make. So like not to give you homework. Give yourself at least a week. At least a week.
I mean, I think when I have it written down, it's like right after spring break. So
Is when you present.
no, like to practice.
Yeah.
Before.
Maris, hang tight. We'll just start.
But maybe I don't have to do it that day. Like she said, right on it. Yeah.
Like I'm not surprised and you have it
It's
missed a week.
probably the day of the test. I have to practice.
Don't do that.
Yeah.
Don't do that. But like when
I'll
we
let
come
you know
back,
when I email you tonight.
and if you find that there's not enough slots, like send me an email that's like, okay, it didn't really fit with what you have open. That doesn't mean that that's all I ask. And tell me these days and times would be really great. Would one of these work? for you? We'll figure it out.
Yeah, I'm
So I want
looking
you
up
to-
the
I want you to-
slides. I know what to say and stuff. I'm just making it, like last night I was making things look pretty, even though like it's just a rough draft.
Right.
We're still
And you
recording.
can kill, oh, thank you. And you can still work on it over spring break. And then